Thank you for agreeing to participate in this research study. If you are watching this, you will have already answered the preliminary questions. While you watch this video, please feel free to take notes, pause, and rewind as needed. Today I'll be showing you the safety guidelines, procedure, and anatomy surrounding a yellow perch dissection. As in all things, safety comes first. Our specimen is preserved in propylene glycol, 2-phenoxyethanol, and 2-amino-2-ethyl-1,3-propanediol, which can be hazardous if ingested, inhaled, or in contact with your skin and eyes. That's why it's important to wear properly fitting gloves and splash-proof goggles, not safety glasses or goggles with vents. This lab will use the following tools: forceps, dissecting scissors, a straight probe, and an angled probe. The dissecting scissors are designed to cut through muscle and bone, so caution should be exercised. Should you cut yourself, immediately leave your station, wash the cut with soap and water, and use the antiseptic and bandages in your lab's first aid kit. You should also notify your professor, TA, or lab supervisor. With that in mind, let's begin the lab by observing this fish's external anatomy. We have the eyes, which take in visual information, the nares here, that take in olfactory information, and the mouth, which is used for respiration and eating. If you run your probe along the top and bottom jaws, you'll notice a rough texture. Perch have very small and fine teeth. Let's look at the fins. As ray-finned fish, perch have thin fins consisting of bony spines and flexible webbing. These are the dorsal fins, which stabilize the fish and directs them through turns and stops. This is the anal fin, which helps stabilize the fish while swimming. These are the pectoral fins, which control left and right movement. These are the pelvic fins, which are used for up and down movement. This is the caudal fin, which moves from side to side to propel the fish forward. Looking closely, we can trace the lateral line of the perch. This sense organ can detect movement and vibrations in the water, which helps them orient themselves. Here we have the operculum, a bony covering that protects the gills. When we pull it back, we can see the gill filaments and the gill rakers farther back. The filaments are the structures that pull oxygen from the water for respiration, while the rakers prevent larger particles, like food, from passing through the gills. Let's use our straight probe to follow the path that water takes through a perch. Enter the fish's mouth at a slight angle, allowing it to pass through a gill arch. You should not need to force it through. If you cannot see the tip of your probe on the other side, it's probably in the esophagus. Now that we've identified the external structures, we can make our first incision. There are actually several ways to do this, but this method is what's taught by our Dr. Miner at BGSU. First, hook the lower end of your dissecting scissors into the anus and cut a straight line up to the jaw. Make sure to keep your blade angled upwards to avoid nicking any organs. Midway through cutting, you'll meet resistance. This is the pelvic girdle, a bony structure supporting the pelvic fins. You may need to change your grip to help apply more force. As you can see, this dissector is struggling. The process of cutting through the structure without causing any extra damage is the most time-consuming part of this dissection. Once the first incision is complete, return to the anus and begin cutting upward in an arc, stopping again at the jaw. You may find it easier to cut off the pectoral fin and operculum first. You will also need to make additional cuts as you try to pull the body wall away. Let's start by removing the gonad with the forceps. Avoid damaging it, as loose chunks of sperm or eggs will escape and clutter the body cavity. In a living fish, these are released into the water by the thousands to fertilize or be fertilized. As you can see, the anterior end has a green tinge. This is because it was in contact with a gallbladder. Now we have a better view of the internal organs. Your fish is now open, but it may not be pretty. As student dissectors, our cuts tend to be less clean with ragged tissue and nicked organs. Here, the student slit open the intestine, causing waste matter to be exposed. Over here, the student sliced open the heart. Here, there's so much damaged tissue that it's difficult to distinguish from the internal structures. This is what a typical student dissection looks like, so it's important to be able to glean information from such a messy sample. Here's a closer look at the gills. You can see the thin gray filaments and the gill arches they are attached to, as well as the little teeth-like rakers on the inner arch. Here we can see the heart, which pushes blood through the circulatory system. If you look closely, you can see the arteries connected to the gills. Fish's hearts only have 2 chambers, as they only need to process deoxygenated blood, while the gills handle the oxygenation process. Here is the liver, which breaks down stored fat and filters out toxins. It's quite large and often needs to be pushed aside to see other organs. Up here is the swim bladder, a delicate balloon-like organ that can inflate and deflate to control a fish's buoyancy. It is difficult to open your fish without shredding this delicate membrane, leaving an empty pocket behind. Along the spine, we can see the kidney, which filters waste and regulates salt concentration in the body. Following the path from the jaw down, we can find the esophagus, and with a little effort, see how it connects to the stomach. You'll need to move the liver aside to accomplish this. This comma-shaped organ is the stomach, where the most digestion occurs. Take note of how it connects to the intestine and the finger-like projections attached. These are the pyloric ceca, which are theorized to produce more digestive enzymes, though it is not conclusive at this point. Arguably the easiest organ to identify is the intestine, which carries out the rest of the digestive process, absorbing any remaining nutrients and connects to the anus to expel waste. This little green organ is the gallbladder, which stores bile until it's needed to aid in digestion. With a little effort, we can find the spleen, which stores red blood cells and contributes to the perch's immune system. Before we conclude, here's a look at why wearing properly fitting gloves is so important. As you can see, these gloves are too large. The extra latex at the end of each finger obscures the organs and makes fine manipulation more difficult. Also, the extra space around the wrist leaves room for fish tissue and preservatives to touch the skin. Conversely, gloves that are too small would also leave the wrist exposed and may rip under the strain. With that, the dissection is complete. Dispose of your fish and any detached tissue as directed by your professor, TA, or lab instructor. Clean your tools and dissection tray mat with soap and water, then remove your gloves and wash your hands. Feel free to rewind or rewatch this video before continuing to the next page in this module.